How can biomarkers differentiate CFS from other illnesses? Imagine trying to tell apart two very similar-looking trees in a forest. You might look at their leaves or bark to tell them apart. That's similar to how scientists are trying to distinguish myalgic encephalomyelitis or chronic fatigue syndrome from other illnesses using biomarkers. Biomarkers are biological clues in our blood, tissues, or other samples that can tell us what's happening inside the body. Right now, diagnosing ME or CFS relies mostly on symptoms like fatigue, sleep problems, and pain. But these symptoms can overlap with many other conditions, making diagnosis tricky. That's where biomarkers come in. They could provide an objective way to identify ME CFS and tell it apart from illnesses with similar signs. Researchers have been discovering unique molecular and immune signatures in people with ME CFS. For example, studies have found specific patterns of cell-free RNA in the blood. These are tiny fragments of genetic material that float around in our blood and can reflect what's happening inside cells. In ME CFS, certain genes related to immune function and energy production are more active or less active compared to healthy people. For instance, increased levels of genes like IL-18R1 and CCR7, which are involved in immune responses, have been observed. Also, altered mitochondrial RNA transcripts suggest that energy production in cells might be disrupted specifically in ME CFS. These differences could serve as biological fingerprints for the illness. Autoantibodies, which are immune proteins that mistakenly attack the body's own receptors, have also been linked to ME CFS. Elevated levels of antibodies against receptors like beta, 2-adrenergic and M4-acetylcholine receptors have been found in some patients. These autoantibodies seem to correlate with how severe the illness is and with viral activity, such as reactivation of viruses like human herpes virus 6. This immune profile is not typical in many other illnesses, making it a promising biomarker for ME CFS. Another exciting area involves tiny molecules called microRNAs. These small RNA pieces help regulate gene activity. In ME CFS, specific microRNAs like ANDMIR30 show different levels compared to healthy individuals. These differences might reflect underlying changes in how genes are turned on or off, giving us another layer of biological information to distinguish ME CFS. Blood tests looking at metabolites, which are small molecules involved in energy and chemical processes, also show promise. In ME CFS, metabolomic studies reveal abnormal patterns, especially in pathways related to energy production. This supports the idea that mitochondrial dysfunction, problems with the cell's energy factories, is a core feature of ME CFS. These metabolic signatures can help differentiate ME CFS from other illnesses with similar symptoms, like depression or fibromyalgia. Scientists are also using advanced computer techniques, like machine learning, to analyze large datasets from many patients. By combining different biomarkers, these algorithms can improve accuracy in telling ME CFS apart from other conditions that cause fatigue or immune issues. This approach is helping move toward more precise, personalized diagnosis tools. However, it's important to note that despite these advances, no biomarker has yet been officially validated for routine clinical use. Variability among patients, differences in study methods, and overlap with other illnesses make it challenging to develop a single definitive test. Currently, doctors still relying on clinical evaluations, but ongoing research aims to change that. In the future, we might have blood tests or molecular assays that can objectively confirm ME CFS, helping doctors distinguish it from other disorders more easily. This could lead to earlier diagnosis, better understanding, and more targeted treatments. For now, the hope is that these biological clues will become part of a comprehensive diagnostic approach, improving care for those affected. To sum up, biomarkers can help differentiate ME CFS from other illnesses by revealing unique immune responses, energy production issues, and molecular signatures. These discoveries are still in development but hold great promise for making diagnosis more accurate and less subjective. As research continues, we're moving closer to having reliable biological tests that can support patients and clinicians alike. Remember, always consult healthcare professionals for personalized medical advice, as individual experiences with ME/CFS can vary widely.